Greetings to everyone. My name is Estefany Paredes. I'm presenting the poster Metataxonomics of the Microbiome associated with symptomatic and asymptomatic banana plants in Fusarium Wilt infected fields. Ecuador is the main exporter of bananas worldwide, and its banana production is at risk due to Fusarium Wilt, a lethal disease caused by the soil-borne fungus Fusarium moxisporum special form cubense tropical race 4, present in the border countries Peru and Colombia. Therefore, it's imperative to keep this fungus out of Ecuadorian territory, to implement biosafety measures, prevention and control strategies. Improving soil health and enhancing soil suppressing capacity can be attained by enriching it with indigenous antagonist biological control agents to identify microorganisms with promising suppressive action against FOG Tier 4. We aim to understand the composition and distribution of the symptomatic and non-symptomatic Gros Michel banana from fields infected with FOG race 1 using functional metagenomics based on high-throughput sequencing. This is the first study in Ecuador that presents a comprehensive view of the fungal and bacterial microbiota bananas affected with FOG race 1 and its changes based on symptomatology under field conditions. Our study will aid to generating new perspectives from the sustainable management of Fusarium Wilt. The method began with the extraction of DNA from three types of environment: the pseudostem, the rhizome, and the cell from the rhizosphere. The DNA was used to library preparation considering the 16S subunit gene for bacteria and the internal transcript spacer region 2 of ribosomal RNA for fungi. We used Illumina MySeq amplicon sequencing to obtain individual FASTQ files per sample. Next, we run a complete pipeline, bioinformatic pipeline containing the points described. Following, we perform an exploratory analysis of the microbiome and a microbial ecology analysis, and also an statistical analysis. As a result, we acquired 5 million input reads for 16S and 4 million for ITS2. Unwanted eukaryotic taxa were eliminated, and uninformative samples were filtered and outliers also were eliminated, obtaining a final number of 600,000 bacterial ASVs and 3 million fungal ASVs. The bacterial taxonomic profiles reflected that the soil of the rhizosphere is the most diverse environment. The heat map groups in base of environment in the figure number 1 reveals that the main bacterial genera found in asymptomatic plant tissues were Pectobacterium, Enterobacter, Bacillus, and on the other hand, Klebsiella and Cousaconia were the most found in symptomatic tissue. In the rhizosphere soil, the genera Candidatus ueobacter and Gaelia were the most abundant in asymptomatic samples, in Nitrospira and broader rhizobium in symptomatic samples. Bacterial heat trees show that the members of Bacilli and Gammophytobacteria classes, especially genera from Enterobacterales order, Pectobacterium, Enterobacter, Klebsiella, Bacillus, Paenibacillus, and Pseudomonas, were significantly present in asymptomatic samples. Also, ASV with different abundance were identified in asymptomatic samples, are shown in table number 1, which was verified with an Permanova analysis showing significant differences for the bacterial community based on symptoms. The principal fungal genera found in the microbiota were Ascocita and Anthrodia, both in symptomatic and asymptomatic samples. The presence of Fusarium moxisporum significantly higher in symptomatic samples, as you can see in figure number 4, indicates that the fungus did not colonize the plants, although it was present in all of the environment. The analysis of microbial ecology reveals that the bacterial community of the rhizosphere soil is more diverse. It obtained the highest taxonomic richness and highest scores in the diversity index. So the principal coordinate analysis showed a clear separation of the soil bacteria population from the plant tissue. As conclusions, The members of the Enterobacterales order and the Bacilli class occur in greater relative abundance and differential abundance in asymptomatic plant tissue samples. Fusarium acisparum and Rhodotula macillafinosa were significantly more abundant in symptomatic samples in the pseudostem and rhizosphere respectively, and the members of the Ceratobacilliaceae family in asymptomatic samples from the rhizosphere. We would like to acknowledge and thank the Corporación Ecuatoriana para el Desarrollo de la Investigación y la Academia, CEDIA, for financing the project CEPRA 14-2020-06 Fusarium Initiative. Thank you very much.